I have already told that I will teach something new in the next subsequent lectures, and I have also conducted one poll for you on YouTube, my YouTube post, and you people have voted for the uh, to for the topic like different kind of vectors used in cloning. So today I'm going to discuss that topic only because it has got maximum votes. Likewise, I'll subsequently give different topics by voting wise, and whatever vote you will give, the maximum vote will be there in the poll. I'll teach that topic. So first of all, we'll see what are the objectives we are going to study here about the cloning vectors. So after the end of the this lecture, you will know what is a cloning vector. You will know about what is a cloning vector. So you'll know about what is a cloning vector, why cloning vectors are used, what is the history of the cloning vector, and what are the features of the cloning vector, and what are the different types of the cloning vector, like plasmids are there, bacteriophages are there, cosmids are there, bacterial artificial chromosomes, that is also called BACs, then yeast artificial chromosomes are there, it is written as YAC, not BAC. Then human artificial chromosomes are there, retroviral vectors are there, which are used in cloning. So what determines choice of vector, how you choose that I have to use a particular vector for particular cloning. So what is that choice, how do you make that choice, and vector in a what are vectors used in basically in the molecular clone gene cloning. So you'll see about that. Now first of all, we'll see what is a cloning vector. The molecular analysis of the DNA is possible by the cloning of the DNA. So basically, when you have to do any molecular analysis of the DNA, you have to clone it inside a vector. This is the first step. So the two molecules are required for cloning, that is DNA to be cloned, and one is cloning vector. So suppose this is your vector. Vector is generally circular in shape and it is present inside the bacteria. So bacteria contains plasmid and it has its own chromosome. So this circular piece of DNA is called vector, and this vector is basically used in cloning, and so that's why we need a vector along with a gene of interest. So this is found mainly in the bacteria. So we require two things for the cloning. First one is DNA to be cloned. Which is our gene of interest. Another one is cloning vector. This is like plasmid, which is obtained from the bacteria. That is called cloning vector. Why it is called vector? Vector is any substance which is used to carry the DNA. That's why it is called vector. In biology, in biotechnology, vectors are basically carriers which carry the foreign gene of interest inside E. coli or bacteria or any mammalian system. That is vector. So cloning vector is a small piece of DNA taken from virus, plasmid of the cells of the higher organisms, or that can be stably maintained in an organism into which a foreign DNA fragment can be inserted for the cloning purposes. Most vectors are genetically engineered, so they are genetically engineered to accommodate different kind of elements like selectable marker gene, origin of replication are there, different kind of origin of replications like low copy, it can be low copy, it can be high copy number, different kind of selectable markers like antibiotic resistance will be there. Then we engineered sites are there that are called MCS, multiple cloning sites, which are required for the cloning. It is having multiple kinds of restriction enzymes so that we can use particular restriction enzyme and we can clone our gene of interest. So most vectors are genetically engineered according to need. The cloning vector is chosen according to size and type of the DNA to be cloned. Uh, the cloning vector, choosing cloning vector is very important because the size and type of DNA will decide your cloning vector. Suppose you want to clone a very big chunk of DNA, you will choose a bacterial artificial chromosome because it can accommodate very high amount of DNA inside back. So at that time your choice will be back. Now I'll see what are cloning vector. So basically a cloning vector is a carrier which carries your gene of interest inside a host like E. coli or any mammalian system, any mammalian cells. So vector contains, basically it has features that allow the convenient insertion or removal of DNA fragments in and out of the vector. So suppose this is a vector and you cut it with a particular restriction enzyme, that is R1 and R2, and this is gene of interest. So you can either cut it and you can either clone it inside this vector. So that's why it is called insertion or removal of DNA in and out of the vector with the help of the restriction enzyme. And suppose you want to insert it inside the vector, then you will use ligase, and you just want to cut it, you will just use with the help with the help of the restriction enzyme. After a DNA fragment has to be cloned into a vector, it may be further subcloned into another vector. Subcloning is very important. Most of people don't understand what is the meaning of subcloning. Subcloning means, suppose you have a vector here and it contains a particular gene, and suppose this gene is just here to express a particular viral protein, and you want to somebody wants to just use it for another purpose, like somebody wants this gene to be expressed expressed and this time it is not expressing in this vector because this vector is not expression vector. And suppose another vector is there and that vector is expression vector, I call it E, just for the sake of understanding. Suppose we cut this with the restriction enzyme R1 and R2. R1 and R2 can be any enzyme like EcoR1 or HIM3, and you just clone it inside this expression vector, this gene, this red one. So this step of cloning from one vector to another vector is called subcloning. Okay. So after a DNA fragment has to be cloned into a cloning vector, it may be further subcloned to another vector designed for more specific use. Like I took an example, like this is a cloning vector only for cloning, so that it will only maintain the copies of this DNA, chunk of DNA. But if you want to express it, you have to clone it inside another vector, that is called expression vector, and this is called what? This is called subcloning. So this is called subcloning, means you are just cutting it and putting it inside another vector from one vector, that is called subcloning. Now we'll see why cloning vectors. Now why cloning vectors are required? Because I have already told that this maintains the copy of the DNA inside the host. It does not express anything. So cloning vector is used as a vehicle. It is mainly a vehicle to artificially carry the foreign genetic material into another cell where it can be replicated and expressed. Okay. For expression mainly we use the expression vectors. It is written here expressed, but this is not totally true. For cloning vectors, it is only for the cloning, not for expression. Expression vectors are different. So basically it is used to amplify single molecule. Amplification is correct because cloning vectors are mainly for the amplification. A single molecule of DNA into many copies. Cloning vectors are DNA molecules that are used to transport cloned success sequences between biological host and the test tube. Okay.